Hello everyone, welcome back to STEM Nerds. I'm Jason. In today's video, we are going to be talking about DNA replication. How does a cell make more DNA when it's trying to make another cell? We're going to talk about the enzymes involved, the process. Hope you enjoyed the video. So I think it's really important before we actually get into the process of DNA replication, let's just make sure we're on the same page, cover a few definitions here. A gene is a functional unit of the genome. It codes for a protein. The genome consists of all of the genes for a single organism. It makes up all the DNA. When it comes to how we study the genes, well, there's two main ways we can do that. One is genomics, and in that, people who study genomics are looking at how the genes are found inside of our chromosomes, if it's human beings. So location of genes, how they interact with one another, how they're controlled, where genetics is mostly concerned about how genes are passed down from one generation to the next. Now, it is important to understand there's different types of DNA depending on the organism. We human beings, we have linear DNA that's broken out into 46 different chromosomes, and different animals, different organisms will have a different number of chromosomes. We also have in our mitochondria, mitochondrial DNA that we primarily get from our mother. However, bacteria, they do things a little bit differently. They actually have circular chromosomes, a single circular chromosome made of DNA. And if you remember from a previous video, a lot of bacteria also have plasmids, which are small extra chromosomal pieces of DNA that are usually not necessary under normal conditions. All right, so here is the structure of DNA. You can see on the right-hand side, we have what is known as a double helix. And it's kind of like a winding staircase. And the DNA is made up of what we call nucleotides. And the nucleotides are shown on the left-hand side. And you can see on the far left-hand side, we look at the top where it says 5' end, way in the upper left-hand corner, we're going to see we have a phosphate group. Then we have a sugar, that's the gray area. And then we have that thing labeled T, that's the nitrogenous base. And then you can see underneath that, we have another nucleotide, same thing, phosphate, sugar in the center, and then we've got that thing labeled G, that is a nitrogenous base. And so what we can see here is that when it comes to nucleotides, we either have a nucleotide that is a single ring or a double ring. And we're going to notice on the right-hand side, closer where it says the 3' end, something weird is happening, and that is the nucleotides look upside down. That is not a mistake. That is the way that they are supposed to be. And so when it comes to DNA structure, we say that the two strands are running anti-parallel. It's kind of like a two-lane road where the cars are going in opposite directions. It's the same thing that we have here in our DNA, and that's what partially contributes to that double helix. You're going to notice something else. You're going to notice that in all cases, we have a single ringed structure. So at the very, very top, we have that letter T, and it is bound to a double ring structure. And that is the way it is throughout all of the DNA. For every DNA strand, we have a T, which stands for thymine, that binds with A. You can see that at the top, and the next one down, we have a G, which is that double ring structure that is bound to C. And so we can say that when it comes to nucleotides, we're making that double-stranded DNA, that A's and T's go together, and C's and G's go together. The next thing that you're going to notice, again, that single ring structure, so the single rings are your T's and your C's. Those are called pyrimidines. The A's and the G's, that double ring structure, those are called purines. And so you always have to have nucleotides across from one another. You have to have one purine and one pyrimidine to get a nice, solid double ring structure. And if we ever run into a situation where we either have two purines or two pyrimidines bound to one another, that creates a structure in the DNA that just doesn't work. And usually there are enzymes that can sense when that happens and correct those mistakes, something that we'll talk about a little bit later. And so we have these 4 nucleotides that are really identified by the nitrogenous base because the phosphate and the sugar in DNA is always going to be the same. So T for thymine, you have A for adenine, G for guanine, and then C for cytosine. You're going to notice something else about the structure of DNA, and that is we have these numbers, and I've kind of alluded to them already. We've got this thing called a 5' end and a 3' end. And those numbers have to do with the carbons associated with that gray sugar in the center. And basically what we're saying is on the 5' end, it's the 5th carbon in that gray sugar that is exposed with that phosphate group. Now you're also going to notice we have a 3' end. Now this one is not as obvious because we have the third carbon. And what's not shown here is there is a hydroxyl group, an OH, that's hanging off that 3' number 3 carbon on that sugar. Again, it's not shown here, but it's just a way so that we can orientate ourselves to what's happening in those nucleotides. And these numbers are going to be really important for us when we start talking about the process of replication. So it's important to note replication is happening when a cell needs to make another cell. Replication is not happening at any other time. And so what I want to point out here with this picture is imagine this is a part of either a linear chromosome or a circular one. It doesn't really matter. But the main point here is that to get replication of DNA, you have to separate the 2 strands of DNA. And that usually happens at an origin of replication. And so there's a spot at which the enzymes we're about to talk about are going to recognize, okay, here's the beginning of where we're supposed to replicate, and then the replication can happen bidirectionally. It can go in 2 directions. It can go both to the left and to the right. And so again, what I'm showing you here is the starting point for replication. Don't worry about the enzymes right now. We're going to discuss those in the next slide. All right, so this is really the meat of this video. This is where we're going to spend the vast majority of our time. What we're looking at here is what is known as the replication fork. This is 1/2 of that bubble I just showed you. So if you will let me go back 1 slide. And so we're looking at the replication fork right here. What we're seeing, if we go back to where we just were, we're looking at the left-hand side of what this replication bubble is. All right, so we're looking at the left-hand side. So now we go to the replication fork. And what we're going to do here is we're going to identify what are the enzymes involved, what do they do. Generally speaking, at least conceptually, the order in which they would need to appear here in order for this thing to work. But a lot of times, well, especially in these more introductory type courses, we're showing these enzymes working individually. And in reality, we have what's known as a replisome, and a lot of these enzymes are semi-connected in some way so that everything is happening mostly simultaneously. This whole process is happening very, very quickly. But for simplicity, so that's easier to learn, we're actually showing these enzymes kind of off on their own, and we're going to look at this as a stepwise function and not them just working all together all at once. Okay, are you ready? So here we go. Replication fork. First thing we're going to need in order to copy DNA, and again, the reason why we're copying, the cell is copying the DNA, is to make another cell. All right, so step 1, we're going to need this enzyme called helicase, and that is the green enzyme. Almost looks like a guitar pick for those of you who play guitar, but that helicase right there, it has 2 jobs. It's going to melt or separate the 2 strands of DNA, and it's going to unwind that double helix, and so we get a more linear portion of the DNA where DNA replication is happening. And so we need that to happen first, okay? We have to be able to separate the DNA, and we have to be able to unwind it before replication can happen. So helicase, thing number 1. Now this second step, it has a few different names depending on whether or not we're talking about eukaryotic or prokaryotic replication. But in either way, step number 2 is going to involve something called DNA gyrase / topoisomerase. And that is that little pink circle that you see there. So imagine you have something that is naturally and normally wound up in a double helix, kind of like that winding staircase. And then you have an enzyme that twists it so that it's basically linear and separates the 2 strands. Well, what's the thing going to naturally want to do? It's going to want to fold right back up on itself again. But we can't have that happen. We just spent a lot of work trying to unwind this thing and separate the strands. And so what happens here is the gyrase / topoisomerase is going to relieve some of the tension in that double helix, basically by cutting one of the strands, and that's going to relieve enough of the tension to hold this thing open long enough for replication to happen. The other thing that's going to happen, especially so in eukaryotic organisms, are what is known as single-stranded binding proteins. Yes, this is another structure that is going to allow the DNA, the double helix, to stay open so that we can begin the replication process. All right, step 3, now that we've got this thing open, we've separated the 2 strands, we've got some security there in terms of the gyrase doing its job, now we need a starting point for replication. And that's going to be done by an enzyme called primase. What primase does, this is going to sound a little strange at first, but primase is going to lay down a little RNA primer within the DNA. So we're mixing right now. We're mixing RNA and DNA together temporarily. So we're going to have this really short primer that is going to eventually tell DNA polymerase III where to begin the process of replication. It turns out DNA polymerase III cannot just attach itself to the DNA and just start, right? It's not a go-getter. It's not a self-starter. It needs a starting point. And the purpose of the primase is to tell the DNA polymerase, hey, hang on to here. It's kind of like a temporary railing. Just hold on right here and then begin the process of replication. So everything that has happened so far, I like to call the preparation for replication, because we haven't actually replicated anything. We haven't inserted a single nucleotide at this point. The primer is there, but no replication has begun until we get to the next step, and that next step is the addition of DNA polymerase III. All right, so DNA polymerase III, that is the blue enzyme that you see there. It is labeled, and DNA polymerase III is responsible for the actual replication of the new strands of DNA. Now, there's a couple things we need to know about DNA polymerase III that is hugely important. One, DNA polymerase III reads the template strand. So the DNA that was already there, it reads the template strand 3' to 5'. Those numbers I mentioned earlier, they're really, really important. Take a look at this picture. On the far right-hand side, you're going to see the 3' end at the top. That was our template strand. And so the DNA polymerase started at the primer, and it's working its way from the right-hand side of your screen where it says 3' in the upper right-hand side, and it's working its way towards the left until it gets to the 5' end of that top DNA strand. So DNA polymerase always reads 3 to 5, but remember what we said a couple slides ago, and that is DNA polymerase, if it's reading 3 to 5, the DNA strands are anti-parallel, which means, if you look at the picture, it's manufacturing the new strand of DNA 5 to 3. And again, we see that in the picture on the far right-hand side. The newly synthesized strand is at the 5' end, and it's going to be finished at its 3' end. Now you're going to notice there is a second DNA polymerase, and that is DNA polymerase I. We have to do some cleanup, and that cleanup is we laid down a small RNA primer. Remember, that was the structure that the DNA polymerase III used as a starting point for replication. Well, we can't just leave the RNA primer mixed in with our DNA. That would be highly unstable, and later on it would be an area where we would have no ability to read that DNA and eventually make proteins. And so DNA polymerase I comes in, removes that RNA, and replaces that RNA with DNA. And so it's cleaning up those little stretches of RNA that we've left behind. DNA polymerase I is going to have a lot more work to do on that bottom strand, and we're going to get to that in a second. Right now, we've primarily focused on that top strand because it's kind of simple. But that bottom strand, if you've been looking at it, you'll notice it is a mess, and we're going to address that in just a second. But DNA polymerase I, removing the RNA primers, replacing the RNA with DNA. Last enzyme we're going to talk about, and that is ligase. You're going to see ligase down at the bottom right. DNA ligase, it's that yellow enzyme. Now here's what ligase is doing. So we have this bottom strand, and up to this point we've kind of ignored it, but we really can't because we have to explain what's going on here, why there just seems to be so much more happening in this bottom area relative to the top. It seems kind of messy. Well, one, remember, DNA polymerase can only read a template strand from 3 to 5. Second important point, our strands, our initial template strands were anti-parallel. We were fine at the top strand because in the upper right-hand corner, our template strand was over there at the 3' end, and DNA polymerase III could simply read 3 to 5 till it gets to the 5' end on the far left-hand side. But the bottom template strand is running the opposite direction. Here's our issue. Our helicase, our little green guy, that helicase is working its way to the left of your screen. On the bottom strand, look what's happening. To the far left, that's the 3' end for the bottom template strand. DNA polymerase can only read 3 to 5, and so what's happening is as that replication fork is opening up, We're getting enough space so we can get a primer in there, and then DNA polymerase still has to read 3 to 5. And so on the bottom strand, it has to read from left to right. It has to go towards the right-hand side. And so as that bubble, as that replication fork opens more and more and more and more, and we can continue to get more primers in there, the DNA polymerase is going to the right, and it's basically just going until it hits the primer that was already laid down in front of it. So we have a little bit of a mess. And that mess means we're going to have to remove a lot more primers because we just keep sneaking in a primer. DNA polymerase goes to the primer that was in front of it. Another primer, and it goes and it's just reading in small stretches. We call this bottom strand the lagging strand and the top strand the leading strand. The leading strand is the strand in which the DNA polymerase is following the helicase, in which case it's a nice continuous strand. So the leading strand is also known as the continuous strand. On the bottom, what's typically known as the lagging strand is discontinuous because our helicase and our DNA polymerase are running in opposite directions. And so the DNA polymerase has to wait for a primer to sneak in there before it can attach, but then it has to go the opposite direction of the helicase and it's just hitting the primer in front of it. Okay, back to our ligase. So what our ligase is doing is when the DNA polymerase goes until it gets to the nucleotides in front of it, it cannot close that last gap. And we call those chunks of discontinuous replication Okazaki fragments. And they all have to be connected together. In the backbone, those Okazaki fragments all have to be connected together. DNA polymerase cannot do that, and so that is the job of ligase. Ligase connects the Okazaki fragments together. This is confusing, I get it. And if you're really trying to understand this, you may have to really visualize this and have to watch this multiple times. Okay, so to summarize, the top strand looks nice and pretty, nice and seamless, and quite frankly, a whole lot easier to deal with because our DNA polymerase started at the primer in the upper right-hand corner where DNA polymerase 1 now is removing that primer. But it would have started over there in the top right. Because it can read 3 to 5, it's following the helicase all the way to the 5' end of the top strand. On the bottom, we have an anti-parallel situation, and so what's happening is the helicase is going to the left. All the DNA polymerases have to go to the right. And so there's a little patience involved. A primer comes in, DNA polymerase 3 comes in, and then it goes to the right-hand side because it still has to read 3 to 5 and manufacture 5 to 3. As the replication fork opens more, we can get another primer, another DNA polymerase, and it can move to the right as well until it gets to a point where there's already nucleotides there. So if you really want to see something interesting, take a look at this. This is going back to the replication bubble. And what you're going to notice is these helicases, there's actually 2 of them, one on the left-hand side, one on the right, and they're going in opposite directions. And so the leading and lagging strands at the point of replication actually flip depending on what side of the replication bubble you're looking at, the replication fork on the left or the replication fork on the right. All right, so this process is going to happen in bacteria. If it's a circular chromosome, it's going to start at the top, work its way to the bottom. When they meet, replication over. If it's a linear chromosome like we have in most eukaryotes, there's going to be multiple origins of replication throughout a linear strand of DNA, and that replication is just going to happen until it hits a replication fork that has started somewhere else at a different point of origin, or until it gets to the end of the DNA. All right, everyone, so that was replication. Again, this is a simplified type of replication. When you take molecular biology classes, when you take cell bio, we start to add in a little bit more complication there. But as always, thank you so much for being here. Hope you have a great week.